So first of all, it's hard to follow up a talk like you just gave, but you gave me, I was looking for a band name, and I think Social Fluids is probably the perfect name for a band. Anyway, so thank you to the organizers to let me present this research that was funded by HFSP. Earth is famous in this part of the galaxy for two things. Life, we don't need to explain that any further, and oxygen, molecular oxygen, O2. We don't really appreciate this because we have 20% of it in our atmosphere, but globally and also universally, O2 is really rare. Looking at the universe in total, for example, diamonds are much more common than O2. So if you want to give your partner something really precious, forget about diamonds. O2 is the much more precious material. Why is it so precious? Why is it so rare? It is because it's a powerful and very reactive oxidant. Two oxygen atoms do not want to be in one molecule because they steal each other's electrons. So O2 is one of the most dissatisfied molecules, and it really wants to be rather a CO2 or a water molecule or a sulfate, which are some of the rather satisfied molecules. In turn, that means O2 is extremely difficult to make, and that's why it's so rare overall. So there's only few natural sources of O2 on Earth and potentially or likely in the universe. And that is photosynthesis is the major process. We heard of this a couple of times in this meeting already. It's a biotic process where water molecules are split to release oxygen, and the electrons that are released from that are used to fix carbon dioxide into biomass. There's a few other more obscure ways of producing oxygen or O2. That's the radiolysis of water. It's an abiotic process that is rather minute, where water molecules are split into hydrogen and oxygen by radioactivity. Then we have the electrolysis of water via lightning and other also rather rare events. That's also a minute and abiotic process. Then about 30 years ago, someone found that there is actually a light-independent way of producing O2 that is carried out by microbes. And it was thought that this is also kind of a freak metabolism that is not really relevant on a global scale. So our idea of the distribution of O2 on Earth was as follows. Any ecosystem that is sunlit and that is in close contact with a sunlit environment contains O2. And strict aerobes like us. And in turn, environments that are permanently dark and isolated from surface processes like the deep subsurface are thus devoid of oxygen and organisms that strictly require O2. Now, this is the idea that I had in mind when I started my second postdoc in Calgary, in Canada, and I was looking at groundwater microbiomes, a really large-scale assessment of these microbiomes. I looked at 14 major aquifers, 87 monitoring wells over the course of 4 years, over 160 wells for the geochemistry and 140 wells for the microbiology. And I categorized these waters based on age. That was something that made sense to me as a microbial ecologist. So what you see here, the yellow dots are rather young groundwaters, shallow groundwaters. Then we have intermediate aged ones in red, and old groundwaters in blue. They have sulfate, and the ones in purple, they do not have sulfate. Just as a reminder, 210,000 square kilometers. You're a mainly European audience. I don't need to remember what square kilometers are, but that's about 3 times the size of Ireland. And so this is really a huge, huge study that is quite representative of the groundwater ecosystems in that part of Canada. So surprisingly, though, what I found was that many of the really old and deep waters. I don't know if this is working now. Yeah, here. If you look here, this is well depth, and still at 200 or 230 meter deep wells, we have substantial amounts of oxygen, dissolved oxygen. In particular, in these old waters, we found a lot of oxygen, and that was and strictly aerobic microorganisms, and that was kind of an anomaly. I didn't know what to do with it. We ran a lot of tests. Of course, the first thing that I thought was I contaminated all these samples with air from the atmosphere. To really figure out if this oxygen is a contamination or if it's something else, I did oxygen isotope analyses. And just to recap the high school chemistry, there's 3 isotopes of oxygen, 16O, 17O, and 18O. 16O is by far the most abundant. 99% of all oxygen atoms on the planet and also in O2 are the light isotope. And then we have the 18O, that's about 1%, and 17O is really rare. If I would close the doors here in this room and we all respire the oxygen because we're aerobes, the oxygen content in the residual air would go down. And as organisms, as life, we tend to prefer the lighter isotopes. So we would remove a fraction of the lighter isotopes from the air, and the residual air in this room would get a heavier isotopic signature than before. Okay, so respiration, now going to this graph here, what we saw was, this is a graph that shows the heavy isotope, so the ratio of delta 18O2 versus the ratio of O2 and argon. Argon is a noble gas that stays constant, so respiration would be found on somewhere on this part of this diagram. If you just leave a water, basically a glass of water open here, it would equilibrate with the air, and you would find a value of somewhere on 24, about 24 per mil, and a ratio of 20 because we have about 1% argon in the air and about 20% of oxygen. Now, if you have respiration, as I just said, the residual air would get heavier, and oxygen is respired, argon stays the same, so you would find the values somewhere around here. What we saw, however, was we found oxygen that was lighter than atmospheric and had a higher concentration than atmospheric. So this cannot be just a contamination of air. There must be an in situ production of oxygen. And of course, it cannot be photosynthesis, so it must be something else. So we had a bold idea. What if O2 is widely produced in ecosystems that are permanently dark? And this is the bold idea that HFSP was bold enough to finance and to support. And I was asking 2 other extremely talented researchers, Beate Kraft from southern Denmark. She's a microbial ecophysiologist working on organisms that do this process. And then Jordan Hemingway, he's at the ETH Zurich. He's an oxygen isotope geochemist. Now we also have postdocs. Tatiana is actually here. She had an amazing poster at the first poster session. I hope you were all there. She developed a method to detect oxygen isotopes in methanol, which is a very, very important tool that was missing in the toolbox. Since everyone is showing group photos, I'm going to show one too. This is probably my favorite group photo I've ever taken in my life. This is us in 3 kilometers depth in a South African gold mine to sample one of the oldest accessible ecosystems, microbial ecosystems on the planet. If I have a little time at the end, I would like to show you a video of what we do there. So the first things that we did, and I'm of course not showing all of the things that we have found out, but I want to show you a little bit on the oxygen that is produced by these bacteria, the isotopic signature, and on some of the major pathways. So the 3 major pathways that I referenced already to is the chlorite dismutation. Here's chlorite is dismutated by the chlorite dismutase into chloride and oxygen. And then we have the nitric oxide, 2 pathways of nitric oxide dismutation. One is 2 nitric oxide molecules are dismutated by the nitric oxide dismutase into N2 and O2. And then we have a third one that actually Beate Kraft just recently discovered, where we have 4 NOs that are turned into 2 laughing gas molecules, so 2 N2O and 1 O2. The gene, the pathway for this is still unknown. So what we did was we looked at the genes that are known so far. And just to tell you that all of these are pretty recent. So the chlorite dismutation was discovered in 1996, but the other 2 only in 2010 and 2022. So this is a very new kind of field in microbiology. So going back to the oxygen, now you have seen a similar graph like this, and I don't need to really explain it anymore. Here's the oxygen isotopic signature, and here's oxygen concentration. This is the work by Jordan Hemingway and his postdoc, Carolina. What you see here, again, is the different barriers. So this is respiration. Everything that's here can be sort of, all the dots that are here can be explained by some kind of combination of diffusion, of atmospheric air, and respiration. This here is the diffusion barrier. So diffusion has an effect, can sort of have an effect, an isotope fractionation effect, but only to a point. So all the dots that you see here, and these are all, every single dot is a groundwater measurement from global studies. So this is a reanalysis of global groundwater studies. All of the dots that you can see here can be explained by some version of atmospheric oxygen being diffused and respired, etc. However, 50% of all the groundwaters that we reanalyzed globally fall into this part of the graph, and that can only be explained by an in situ source of lighter oxygen, so lighter than atmospheric oxygen. So I love that. Of course, I was first of all, to say that this is completely serendipitous. I had no interest in working with oxygen as a postdoc, but it really turned into the thing that I do now. And so, of course, I had to read a lot of papers, and I came across this paper here by Vinograd and Robertson from 1982, a science paper, where they look at oxygen anomalies in groundwater, and they have no explanation for it. And they say, we hope that this report will stimulate a systematic appraisal of dissolved oxygen in future geochemical studies of shallow and deep groundwater. And thanks to HFSP, their dream 45 years later now finally is coming true. We find the genes for this process. So this is work by the postdoc in my lab, Emil Vedal. The color codes here, so this is a phylogenetic tree of the gene, and where we found this gene is color coded. And you see, we find genes of this process in marine waters, in fresh waters, in groundwaters, in deep sea ecosystems, in soils and sands. You name it, it probably contains organisms that can do this process. Similarly, and that's in collaboration with an assistant professor at University of Nevada, Las Vegas, Ranjani Murali, we showed that the diversity of these genes is extremely high. Before this analysis, we basically knew this clade here, but we showed that there's major clades of this gene family that we have totally overlooked. So we had 17 phyla that have the genetic capability to perform this nitric oxide dismutation. 12 were added by this analysis alone, and this is just the beginning. So we just made a new tree that just has more again. And we did the same thing for the chlorite dismutase. There's an extremely high diversity of this gene in the tree of life. So to sum up basically this first study was that we really showed that we have indications, either geochemical or biological indications of this process in almost any ecosystem that we looked at, in freshwater bodies, wetlands, in contaminated aquifers, in anoxic aquifers, in hydrocarbon reservoirs, in deep crystalline bedrocks, methane seeps, and so on, and so on and so forth. So this is a very, very widespread process that we did not have on our radar at all. Also, a few years ago, I published a global atlas of subsurface microbiomes, similar to what we heard yesterday, this atlas of ocean microbiomes. And here too, organisms that can do this process, Methallomirabiliota, are extremely abundant in subsurface ecosystems worldwide. So what does all that mean? What are the implications of this for the origins and limits of life? Just over lunch, I talked to I talked to the speaker of the first day. The new kids on the block kind of idea really fascinated me. But then when I said that 50% of all microbes on Earth live in the subsurface, that kind of fascinated you, right? You were like, what? I didn't know that. So we have 10 to the 30 estimated microbial cells on Earth. And about 50 to 80% of those live in subsurface ecosystems that are not connected to sunlight. So it's a huge, it's an extremely large volume of habitable volume. So it is important if they can make oxygen or not. And the other thing is that oxygen has been connected to the evolution of macroscopic organisms. Whenever there's a sort of a jump in oxygen concentrations in the atmosphere, there's a jump in organismal diversity and complexity. So maybe we have totally overlooked complex organisms or habitats for complex organisms in Earth's subsurface. And with that, I wanted to summarize, but let me see what the time says. Instead of the summary, and also the outlook on Mars, we just got a big NASA grant because these metabolisms could be important for life on Mars, and that is thanks to HFSP. We now build on this, and I can't be thankful enough. But what I wanted to show you is, and I think I have a minute or two. Yeah? Okay. What I wanted to show you is this journey to the center of Earth. So this is the Moab Khotsong gold mine in South Africa. It's about 4 times the depth of the Burj Khalifa, so 3.2 kilometers. It's one of the deepest mines on Earth. And we had the chance to go there with the miners to one of the deepest points with all our gear. Here we kind of look like the Beastie Boys, which was, of course, an extra perk for me. We took the elevators. This is the deepest single shaft elevator in the world, and it goes at 60 kilometers per hour, so it's like a rattling cage zipping down there. Sometimes we had to take the ride with the miners that you see here, sometimes on our own. Down there we had to take a train, so we took a train ride another 2 kilometers horizontally into this mine, to then end up at the final train station where we had to walk for another about half kilometer, to finally arrive at this blind tunnel where good people and great researchers before us had drilled a science borehole already that accesses water at 4 kilometers depth that has been isolated from surface processes for 1.2 billion years. And needless to say, there are living microbes in there. It's an autonomous ecosystem likely driven by the radiolysis of water. We find oxygen, we find aerobes, and we have no idea how long they have been down there at 3 kilometers depth. And this is exactly the kind of life we would expect on Mars. And with that, thank you for your attention.
Thank you. That was an excellent talk. I'm sure there's questions.
Just a question about the last statement you made. Why do you think life on Mars would be aerobic at all, when maybe they're not using oxygen?
It's probably not aerobic. Let me go back to the summary. It's probably not aerobic, but it might look like this deep subsurface life. The reason why NASA was interested in that is because perchlorate, the substrate for one of these metabolisms, is extremely abundant on Mars. And perchlorate actually NASA has already the plan to use bacteria to turn the perchlorate into oxygen for human habitation. And so this, together with the abundance and probably the liquid water deep down in the crust, made it sort of interesting enough for them to go for it. But